Welcome to Catherine's tutorial. In this video, we'll learn about the complete concept of Lac Operon. Operon model is to explain the gene expression in prokaryotes, especially in E. coli. So here this model was proposed by Jacob and Monod in the year 1961. It explains the lactose metabolism in prokaryotes. So here we are going to discuss about two mechanisms, that is switch off mechanism, that is a Lac Operon. The other one is switch on mechanism. And before getting into this topic, a small introduction about this Lac Operon concept. It is nothing but E. coli obtained the food source from the human intestine because it lives inside the human intestine. It obtained the food source from the human intestine. They absorb glucose as their food product. Sometime if there is no glucose available, they have to take up the lactose from the surrounding medium, that is inside the human intestine. So when they take up this lactose, they have to go for the process of transcription and translation to synthesize enzymes and proteins for the digestion of lactose. When there is a need of lactose metabolism, the organism will undergo a process known as switch on mechanism, where they switch on the genes to produce the proteins or enzymes necessary for the lactose metabolism. If there is no need, if they have glucose available in the environment, no need to go for lactose metabolism because lactose metabolism is an energy consuming process. So what they'll do, they always go for switch off mechanism, where they utilize glucose if it is present inside the surrounding medium, or if it is not, they'll go for the lactose metabolism. To initiate lactose metabolism, the genes has to undergo transcription and translation, that is known as switch off and on mechanism. We'll just compare these two so that you can able to understand it easily. So this is the DNA which is present inside the E. coli. So this is DNA. Here we have a promoter gene, LAC-I gene, followed by one more promoter, and you have operator gene, followed by the structural genes. You have termination codon at the end to stop the process. Here the same we'll draw here. So promoter region, LAC-I gene, one more promoter, operator, and three structural genes. And here we have a termination codon. So this mechanism is switch off mechanism. Here glucose is present. So during the switch off mechanism, when the glucose is available, the organism no need to undergo the process of lactose metabolism. So this condition is known as switch off mechanism. And we have to learn about one more protein known as catabolite activator protein. So catabolite activator protein. What is the function of catabolite activator protein? This catabolite activator protein site is present before the promoter region. So here you can able to find CAP site. There is one more signal for the switch off and on mechanism. It is cyclic AMP. This cyclic AMP level, that is cyclic adenosine monophosphate level, will decrease when there is glucose in an increased state. Okay, remember the cyclic adenosine monophosphate level will decrease when there is more amount of glucose in the surrounding medium for this organism. So it signals that the organism has glucose in the environment, so there is no need to go for lactose metabolism. This cyclic AMP will bind with this catabolite activator protein and it starts the further transcription process. Okay, so this catabolite activator protein will initiate the RNA polymerase binding with the promoter site when there is a need, and it also suppresses the binding of RNA polymerase to the promoter site when there is no need. Okay, if there is a need for lactose metabolism, the CAP complex will allow this RNA polymerase to bind with the promoter region. So this is known as the promoter region. It allows the RNA polymerase to attach with the promoter sequences to initiate the process of transcription if there is a need. So here there is no need of lactose metabolism. The cyclic AMP level is in a decreased state, so there is no need of formation forming these two complexes, so it is not going to initiate the binding of RNA polymerase with the promoter region. So this catabolite activator protein and cyclic AMP complex will also involve in switch off and on mechanism. So here the organism has glucose complex is not going to initiate the binding of RNA polymerase to the promoter region. So now what happened? LAC-I gene will produce the repressor protein. This repressor protein will bind with the operator region. Okay, remember, imagine this LAC-I gene which produces the repressor protein. When this repressor protein goes and binds with the operator, so this is the operator gene. When they go and bind with the operator gene, the process will not occur. So further process of transcription will not start actually. So what happens here? When the repressor protein binds with the operator region, it will not allow the RNA polymerase enzyme to bind with the promoter sequence. We know already when RNA polymerase attaches with the promoter sequence only, the process of transcription will start. When the repressor protein binds with the operator region, the further process of transcription will not occur. So this complex will not allow the RNA polymerase enzyme to come and attach with the promoter region. So this mechanism is known as switch off mechanism, where there is no need of synthesizing the enzyme necessary for lactose metabolism because glucose is available for the organism. This condition is known as switch on mechanism. So when this switch on mechanism will occur? When there is no glucose in the environment. So glucose is not available for the organism. They have only lactose in the environment. So they have only lactose. So what happens here? This LAC-I gene will produce a LAC mRNA. This LAC mRNA will produce the repressor protein. So the repressor protein is produced. So this LAC-I gene will be going on producing the repressor protein. It is a continuous process. Okay, they are not going to stop according to the need of the food substances, whether it is glucose present or lactose present or absent. This LAC-I gene will go on producing the repressor protein. When this repressor protein goes and binds with the operator region, it shows that they have glucose for their metabolism. When the repressor protein binds with the operator region, it shows that the organism has glucose in the environment. If this particular region, if the repressor protein doesn't bind with the operator region, it shows that there is a need for lactose metabolism. LAC-I gene will go on producing the repressor protein continuously. They are not going to stop, or they are not going to switch on or off the mechanism. When it goes and binds with the operator region, the process will not occur. When they doesn't bind with the operator region, the process will occur. Now the repressor proteins are produced. So this is the repressor protein. What happens here? When there is switch on mechanism, the lactose goes and combines with the repressor protein. It makes the repressor protein to be inactive. Now this inactive repressor protein cannot go and bind with the operator region. Now the operator region is free. It will allow the RNA polymerase enzyme to come and bind with this promoter sequences to continue the process of transcription. That is, they produce a LAC mRNA. This LAC mRNA will undergo a translation process where they produce the enzymes necessary for the metabolism of lactose. So, and one more important thing, this CAP, that is catabolite activator protein, and cyclic AMP complex will be formed. So, when there is no glucose in the surrounding medium for this organism, the cyclic AMP level increases. Cyclic AMP level increases when glucose level decreases. So this initiates for the formation of this complex, that is with the catabolite activator protein, which is present in front of the promoter sequences. So this catabolite activator protein complex will also initiate the binding of RNA polymerase with the promoter region. But when you compare here, the cyclic AMP level decreases when the amount of glucose, when the cyclic AMP level decreases when the organism has glucose. So this will not allow the RNA polymerase to come and they will not initiate the RNA polymerase to come and bind with the promoter sequences. But here the cyclic AMP level increases when the glucose level decreases, so it initiates the cyclic AMP complex with the catabolite activator protein. This complex, which is present before the promoter, that is capsid, this capsid will enhance the combining of this RNA polymerase with the promoter sequence to initiate the further process of transcription. So this is the function of cap complex in case of Lac-Operon system. Now what happens? These Lac will produce beta galactosidase enzyme, which will help in breaking down of lactose, which is a disaccharide which will be broken down into glucose and galactose with the help of beta galactosidase. And Lac-Y produces permease enzyme which allows the entry of lactose into the organism, and Lac-A gene that produces trans-acetylase which will help in transferring the acetyl-CoA group. Switch off and switch on mechanism: when there is a need of lactose metabolism, they undergo process of transcription and translation to produce enzyme necessary for the lactose metabolism. But here in cases they have glucose available in the environment, so they will not go for a choice of producing all those enzymes, which is not necessary because those enzymes are used only for lactose metabolism. Imagine if both lactose and glucose are present. The organism has both glucose and lactose in the environment. Preferably, they will go for a choice of glucose. They will also go on, but at a very slower rate because they doesn't need the lactose to be metabolized because they have glucose in the environment. But because of the presence of lactose, the transcription process will occur, and where there is a very slow rate of producing this proteins or enzymes. By the next video, we'll learn about the next concept in molecular genetics.